Well, our interest is in understanding breast cancer stem cells and how they're regulated. We believe that breast cancers are driven by a small component of cells that have stem cell properties, and that these may contribute to tumor metastasis as well as treatment resistance. It's also becoming clear that the tumor microenvironment is very important in breast cancer growth. What we have done now is to look at how cells in the microenvironment regulate stem cells in breast tumors. And what we find is that as breast tumors grow, we think they actually recruit stromal cells from the bone marrow that are actually stem cells, mesenchymal stem cells. These mesenchymal stem cells home to the tumor, where they communicate with stem cells in the tumor. So we have two different cell types, one a bone marrow mesenchymal cell, the other a tumor stem cell, and they talk to each other. We've now worked out the signaling networks that actually regulate this crosstalk, and they relate to cytokines that are made both by the mesenchymal stem cell and the tumor stem cell. What we're finding is that if we take these mesenchymal stem cells and mix them with tumor cells, the tumors grow much quicker in animals. What's exciting about this is we've worked out the specific receptors on the cancer stem cell that regulate this interaction. And we're figuring out ways to interfere with this signaling between the mesenchymal stem cell and the tumor stem cell. As a matter of fact, there's one particular receptor called CXCR1, which is a receptor for interleukin-8, an inflammatory cytokine. What we find is that when we inhibit CXCR1 with a small molecule inhibitor or with an antibody, we can knock out cancer stem cells. We're really excited because we think this is a potential whole new approach to treat cancers by interfering with the crosstalk between the cancer stem cells and their microenvironment. So what we're now looking to do is to test this approach of inhibiting the chemokine receptors in cancer stem cells and combining that with other approaches like chemotherapy. We've also found that when we treat cancers with chemotherapy, we selectively kill the cells that aren't stem cells. The stem cells are resistant. But what's even more remarkable is that the dying cells that are killed by chemotherapy secrete interleukin-8 as part of their damage response. This actually stimulates the cancer stem cells to reproduce themselves. It's almost like a beehive of angry bees. And what it actually is simulating is a damaged tissue. When a tissue gets damaged, it actually releases these chemokines like IL-8 that stimulate the stem cells in the normal tissue to repair themselves. The problem is when we treat cancers with chemotherapy, the cancer stem cells are being stimulated to grow too. So our approach now in the future is to try to improve the efficacy of treatments like chemotherapy by putting in blockers of this interaction. So we're adding the inhibitors of CXCR1 with chemotherapy, and it looks very exciting that we can really knock out the stem cells in cancers as well as presenting cancer metastasis, which we think is mediated by cancer stem cells.
So it sounds like it's got great potential.
Absolutely. As a matter of fact, the small molecule inhibitor that we're using was actually developed to treat heart disease because it's an anti-inflammatory agent that actually can help prevent heart attacks. So it's already been given to patients. So we may be able to move into clinical trials relatively quickly. Well, there's a very important session now on stem cells and the microenvironment because as I indicated in my own research interest, other investigators are also finding that the microenvironment is crucial and it actually regulates stem cells.